Alright, so welcome to another tutorial. This one was requested a little early, so I'm going to be posting it a little bit earlier than normal. This is going to cover Unit 4's Meiosis in life cycles tutorial. So this is again in Unit 4. It's coming a little bit early, alright guys? We're going to cover as much as I can with this one. Honestly, going by the mastery test, this is actually a really hard test. I'm kind of surprised they covered it in only 20 slides. So I'm going to take a look. We're going to see what happens, and hopefully this all makes sense to you guys. All right. So meiosis. This is different from mitosis. All right. So mitosis is going to be you essentially end up with a cell. No, whatever. They're gonna be exactly the same thing. So you're gonna start with one cell. It's gonna split into two. And it's gonna have the exact same thing, right? With meiosis, it's a little bit different, and it's gonna be a sexual reproduction stage, whereas mitosis is just essentially cloning. So like mitosis, you end up with two exactly same things. Meiosis, you end up with a bunch of stuff. You're not really sure what you're gonna end up with. There are statistics to prove all that. That's Mendelian genetics, which you guys probably will have done earlier in Unit 4 if you're watching this video. So we're going to go over meiosis specifically. Let me see. I'm on page 4 of your tutorial, and I'm looking for important things. Let's see. So there are two types of cells in the body. There are gametes, and then there are somatic cells. So gametes and somatic cells. I've learned how to do this in two colors now. So there's gametes, and then here are somatic cells. We're going to separate it kind of big, just because I can. Somatic and gametes. Alright, and I'm going to switch up the colors just because I can, and whatever. So with gametes, these are sex cells. Alright, they have half the chromosomes of a regular cell. So these have half the chromosomes. I might shorten the word chromosomes to like CHROM, so you can just assume that anything that I do with that are going to be half. So these are body cells. So these are things like your muscle cells or your cheek cells or literally anything that's not a sex cell. So sex cells, keep in mind, include things like the ovary, or the sperm in human beings. Now this changes depending on if you're working with plants and whatnot. They're still called gametes, they're just not called ovary and sperm. So these are specifically for humans, but somatic cells are going to be body cells like muscle cells and cheek cells and skin cells and whatever else. These are going to have the regular amount of chromosomes, okay? So this is just regular chromosomes. Alright. So for example, if a somatic cell has I don't know, 24 chromosomes in a given body, so there's some kind of weird animal with 24 chromosomes. Gametes are only going to have 12. That kind of thing. Okay. Let's see what else they say on here. Oh, yeah, so somatic cells, these are created with mitosis. Created with mitosis. Alright, so all of your skin cells, all of your muscle cells, all of whatever, they're generally the same because if they weren't the same, your body wouldn't work the way it is. Like all of my finger cells, all these cells on my skin, they're all generally the same. They just, they're all cut from the same cloth. They have the same genetic material because no matter where from my body you were to take a DNA print, so you could take it from my hand, you could take it from my tongue or my cheeks or whatever, all of those cells should have the same DNA. Okay, and that makes sense because obviously your DNA decides who you are. So all of your body cells have the same DNA. Gametes, on the other hand, carry different types of things. So like not every sperm is the same, not every chromosome is the same, and so gametes are created through sexual reproduction or meiosis. Right, meiosis. Some people call it meiosis and mitosis. If it helps you separate it easier, go ahead. I'm gonna call it meiosis because I like meiosis and mitosis. It rhymes, so it makes it easier for me to deal with. Cool. So they also, this isn't going to be on your mastery test, but this is kind of like a general knowledge thing you should know. Human beings have 46 chromosomes in all of their somatic cells. So let's get this back on camera. So human beings have 46. So their gametes, our sperm and our ovary, have 23 chromosomes. Okay. So like I said, basically just have the number if you're ever finding the gametes from the somatic cells. I'm on page 5 now and I'm looking over it. It talks about how sex cells form by meiosis and whatever. So we've already actually talked about all of that. But I noticed there are a couple of terms on here called crossing over and tetrad formation. Okay, so let's talk about what crossing over and tetrad formation are. Here's tetrad formation. All right, so tetrads look like this. They have little. I'm gonna use multiple colors for this. Let's use like purple first, and then I'm gonna use like this blue. You guys tell it apart? No, you guys can't. Let me make one of these like a darker purple or something. Tell that apart? Not really. What about orange? Yeah, okay, that was pretty distinct. Okay, yeah. So what happens is these are chromosomal strings. So these are strands of genetics, right? These are genes. So what's gonna happen is they form a tetrad. So they form a tetrad. So what's gonna happen is you're gonna get like one thing, and it's gonna still cross with this thing, and then you're gonna get another thing, and it's going to cross, and it's got that purple thing at the top. I know you guys can't really see this so well on camera. I'm trying to make the purple stand out, but it's just kind of not. But what happens is that this thing with four little stems is called a tetrad. I probably should have used better colors. But and not every tetrad has to be different. It can all be the same color. Sometimes it's like two colors, three colors, etc. It just depends on how you want to represent it. But the point is, it has these four different strands. So one, two, three, four. That's why it's called a tetrad. Tetra means four. Okay, so tetra is one step beyond tri. Right. So that is what tetrad formation is. Crossing over is this process. So when I make them cross over like this, sometimes things flip around. So like you notice I kept the purple and the orange both on top, and I kept the orange on the right, purple on the left. Well, in crossing over, I could easily have part of this purple end up down here. So you could end up with purple down here. Again, a little hard to see, but the idea is you could end up with purple down there, or you can end up with orange down here or something like that. You really never know. They're just gonna mix together, and you're gonna get different types of crossing over. When they cross over, they end up moving some of their genetic material around. Okay, so when things get close together, sometimes you bump them into each other. It's kind of like if you put I put 2 cups of like 1 cup of water, 1 cup of soda, and then I shake them up and whatnot. Chances are some of my soda is going to fall in my water, some of my water is going to fall in my soda. That's just kind of how it happens. This works the same way. All right, so let's go ahead and move on to page 6. Okay, so page 6 breaks down mitosis and meiosis. Wow. I'm going to focus on the differences as a whole. I'm not going to talk about any one of them specifically. I am just going to focus on the differences as a whole. So let me separate this off from the rest of my notes, and let's talk about this real quickly. Let's see. So mitosis has one phase. That's actually really, really important to notice. So mitosis is, and let's get mitosis in green. Let's get, you see the difference? Is blue different? Yeah, a little bit. I'm gonna go with orange because it's very different. I don't really like these colors because whatever. But mitosis, and then we're gonna go with meiosis over here. This marker is also a little thicker, so like my writing's gonna look weird with that one. So sorry. But mitosis and meiosis. All right. So this one has 2 phases. Basically, it's going to split twice. You're going to end up with 4 cells with meiosis. Mitosis, on the other hand, only has 1 phase, and you're only going to end up with 2 cells. All right. And when you do mitosis, you're going to end up with 1 cell becomes 2 cells. I just said that, but I'm going to put it in drawing because, I don't know. You're going to get 1 cell becomes 2 cells, and each of these is going to become 2 cells. So at the end of meiosis, you actually end up with 4 total cells. All right. So that's one of the major differences. And each of these cells here, so each of these cells are diploid. All right, so these are called diploid. I'm going to use a different color for this after one more. So these are both diploid cells. That means that they have the full chromosomes. So like in humans, these have the 46 chromosomes. I'm going to use humans and their 46 chromosomes as our base because it's easiest to understand with an example. So they have 46 chromosomes, okay? Over here, on the other hand, these are all called haploid, which means they have half the normal chromosomes. That means they have 23. This is an example of a gamete, okay? So you have haploids and diploids. Mitosis results in diploids, meiosis is haploids. So if you actually keep this all in perspective, remember I told you guys that somatic cells are created with mitosis and gametes are created by meiosis. These are called sex cells. So meiosis is sexual reproduction. So if you've memorized this list already, you already know the process of meiosis. Or if you've memorized, the somatic list already, you already know the process of mitosis. Okay? So mitosis and meiosis. One is sexual, meiosis is sexual. You end up with haploids or half things. You end up with gametes, which have 23, instead of the regular 46 that you get from mitosis. Alright? Let me see. Oh, here's another major difference. I'm gonna write this in pen just because I don't know, I like pen. I'm kind of getting irritated by how the purple writes. So with meiosis, you're going to end up with genetically different. So this is sexual reproduction. Remember, sexual reproduction causes variation. Okay, because of the crossing over, because of the two different parents that you see here, because of the different colors and the way they mix, you're going to end up with genetically different cells. Genetically different. All right. On the other hand, over here with mitosis, you're going to have asexual reproduction. Remember that a prefix is going to mean not, so it's not sexual reproduction. You're going to end up with essentially a clone. So this is genetically same. Genetically clones. I'm just going to write both. So genetically clones. All right, you're going to end up with two things that are exactly the same. These four, you have no idea. So like that's why you never know which sperm is going to get to the egg. So like this sperm could be blonde, while this one's like brown hair, brown hair, brown hair, but that one guy is blonde. Or like this guy's blue eyes, this guy happens to be brown eyes, brown eyes, brown eyes, something like that. You just never know, and that's why in genetics we have Mendelian genetics where we can figure out like what is the likelihood of this. Let me get all this on camera because that's kind of there we go. Okay, so those are the main differences in the human body. Just remember the difference between mitosis and meiosis. All right. So I know this was kind of a lengthy area, but it is really important to understand the differences between the two. We'll go ahead and move on to page 7. See what we've got here. Now we're going to go over the specific phases of meiosis. So feel free to pause here if you need these notes. I'm going to go ahead and move on to meiosis now. So yeah, feel free to pause, and I'll see you when you get back. But here we go. So let's talk about the different phases, and it's kind of interesting that um, this is kind of interesting the way they represent this. So they're going over the three main phases. Let me see if I'm gonna skip ahead real quick in one sec. All right. I'm going to introduce interphase, but because of the way they produce this tutorial, I think I'm going to go over meiosis 1 and meiosis 2 on the next slide instead. So I'm just going to go over what interphase is. But the idea is interphase is the time the cell is not reproducing. Okay, they're just getting ready for everything. All right. So interphase is essentially like here I have a cell. Nope, you guys can't see that. Let me pull this down. So I have a cell, and it has, one, two, whatever, two things in it, right? So during interphase, this cell starts creating more stuff. So instead of just the 1, 2, it'll end up, like 3, 4, something like that. And then it's going to start to split. So interphase is this very important phase where the cell starts to duplicate its genetic material. All right, so you end up with more than just the 2 chromosomes. You end up with like double that, and then you split. All right, now let's talk about meiosis 1 and meiosis 2 specifically. I'll tell you where interphase belongs in all of this in just a sec. So in meiosis 1, we're actually going to start a new page for this, and I'm probably going to write in pen because I'm going to need the room for this one. We might go back to that page later. But I'm also going to write this one landscape. So this one's going to look a little weird on the camera just because this camera's not really meant for landscape. But that's okay. It'll make sense. So we're going to do meiosis 1 first. Like I said, I'm going to be writing in pen because I just need the finer point to write with. So meiosis 1. Okay. First off, you start off with interphase. Interphase. This is step 1. All right, and I'm not gonna number these. Whatever. I'll write step 1. Please don't get confused by this number. So like I write step 1. This is interphase 1, but when I write like step, I think 6 is probably what it'll be. don't think that that's anything other than just the sixth step. All right, so it's not like interface 6 or anything like that. It's step 6, interface 2, or something like that. So let's talk about interface 1. Okay. Oh, that's weird. I thought I'd be able to click on these to see more, so you guys won't do this. So I'm just going to talk about them from my memory, and hopefully this will work out and help you guys. Let's start with interface 1. All right. In interface 1, it's simple. Your chromosomes duplicate. So chromes duplicate. Chromes double. I'm going to call it double. All right. Generally, it is going to be doubling. I guess technically they could like triple, but that's kind of a that'd be a glitch. So I'm going to get this a little closer than all right. So here's meiosis 1. I'm going to go ahead and move over here to step 2 now. And like I said, all this happens is that your genetic material or your chromosomes double. So let's talk about step 2. Maybe I'll separate them with color or something like that. There we go. Yep. I probably shouldn't have done that, but it works. Here's step 2. This is going to be called prophase 1. Alright? Prophase 1. So with prophase 1, you're going to see crossing over. This is where crossing over happens. Crossing over happens. So whenever you see these kinds of questions, prophase 1, you're going to see all of your genetic material, all of your crossing over is going to happen here in prophase 1. Your tutorial says homologous chromosomes pair and exchange segments. Okay, so that's a good way of saying crossing over. Crossing over is a much easier way to say this, so let's just go with that. Just remember, this is where you're going to start exchanging genetic material around. So if they ask a question like, what improves genetic variation in what stage is responsible for genetic variation, it's probably step 2 where the crossing over happens and it creates like randomization, which is a good thing in sexual reproduction. All right, so that's the main thing in step 2. Let's talk about step 3. All right, step 3. This one is going to be your metaphase. So metaphase is kind of easy because it starts with an M. Metaphase is in the middle, so M for middle. Okay, so it's your middle phase here. And all that's going to happen is that all of your tetrads, so remember your chromosomes that are crossed over and have those 4 things, these things here. Actually, let me color them in a little bit. Or green and this can be purple or whatever. So your tetrads like that. Oh, why do I keep mixing green and purple? I know you guys like can't really see that difference. Whatever. So these tetrads are going to line up in the middle of the cell. Line up in the middle. All right. So metaphase is the middle phase, and they're going to line up in the middle. All right, and you can see all these pictures on your tutorial. I'm just going to give you guys like the text breakdown of all this. I might draw some figures, but I don't do well with art, so move on. Let's take a look at step 4. Alright, so here's step 4. And this is going to be called anaphase. Anaphase, unfortunately, I don't really have like a fancy way of memorizing anaphase. So, whatever. Anaphase. Keep in mind, all these are anaphase 1, so in meiosis 1, it's all step 1, obviously. But anaphase 1, what's going to happen is that your homologous chromosomes, and I'm not going to write chromosomes, but what's going to happen here is your homologous chromosomes are going to move apart from each other. They're going to separate. Okay, so all this means that they're going to be similar. Your tetrads over here, when they've lined up in the middle during, where's my camera? It's kind of weird. Yes, to see that. Okay, so when they're here lining up in the middle, all of your chromosomes and whatnot are going to touch each other and be all lined up close. And then what's going to happen is that they're going to separate out during anaphase into which one should belong to what cell. So if you have twins or whatever, your brother and sister are, or brother and brother, whatever it is, your two twins are going to be very, very different things, and it all depends on their genetics. So it's during anaphase that their individual traits are separated. Alright, so twins obviously can look alike, they can be really, really similar, but it's during anaphase that you decide what traits will be individual to each of the individuals. Wow, that was kind of redundant, but you get the idea. Alright, so let's go ahead and move on. Step 5 is going to be the last step of any meiosis. All right, so step 5 is going to be your telophase. Telophase. All right, and in telophase, you're just going to see this is a big deal. All right, every time you're asked about telophase, this is a cleavage. All right, cleavage furrow. And what cleavage is when a cell is, it has these like whatever these things. This is a cleavage furrow. All right, so you have genetic whatever's. Whew, I just wrote those as X's because I got I don't know lazy or tired or something, whatever. But they're floating around doing whatever they do. But you're finally starting to bend in here, and you're about to start the split. All right, so this is the cleavage furrow, or you start splitting. Start splitting. Okay, so to break, oh my god, I can't spell splitting apparently, but that is what you're doing there. So pretend that says splitting, even though it's kind of awful. But the idea is that you're going to start with interphase, so you're going to end up with more chromosomes here. So you create the material for splitting. That makes sense. You want to start with interphase. This is the very intro phase, okay? So the interphase is the intro phase. It introduces and it starts building up more chromosomes so that you can split. Then your chromosomes, since you've made so many, they're going to cross over and you're going to end up with genetic variation, okay? You're going to end up with various traits. You move on to metaphase in step 3, and that's going to be where they all line up in the middle. So everything you have is going to line up in the middle. And then after you both, or after your cell has put everything in the middle, it's going to be like, all right, well, it's time to separate things. So basically, you want to get everything in a pile before you organize it out. So anaphase, you're going to organize it out into 2 different piles. Now that you've got your pile set up, you can split up. This is kind of like a divorce, honestly. It's kind of awful, but actually, yeah, let's run through this like it's a divorce because I think that actually makes a lot of sense. So interphase is kind of like the early parts of a relationship. So you build up a whole lot of stuff, right? Like you buy 2 cars, you buy, 2 boat. I don't know why you buy a boat, but maybe you have 2 kids. That's really awful. This is a really horrific example, but yeah. So you buy 2 cars, you buy 2 of everything, and whatnot. You buy plates and dishes and whatever else. So that's interphase. You pile up crap, right? Prophase is when all of your crap gets mixed up. So like, yeah, I can't tell my car from your car because sometimes you drive my car and you have your plates and my plates. They're all kind of in the same place. Your dishes and my dishes, they're all kind of in the same place, right? So all of our stuff gets mixed up. Now we're divorcing, so like, well, let's talk about it. This is when you go to the divorce court and you're like, all right, well, this is all the stuff we own in both of our names, all right? So you line everything up and you describe what you have. That's step 3. Then you move to step 4 and you're like, all right, well, step 4, homologous chromosomes separate. I don't know why I didn't write that. Chromes separate, all right? So this is where we separate our stuff. We're like, all right, well, we lined them up in the middle, but now we got to split it up. Like, this is yours, this is mine, this is yours, this is mine. You just create the 2 separate ones. Now that we've got it all separated legally, we can, build our separation and eventually move into 2 cells. So telophase is where we move into those 2 cells or we divorce out. All right, so that is meiosis 1. Meiosis 2 is actually going to follow the similar process. Oh, my bad. I actually skipped the page, but I noticed meiosis 2 is actually on the same page. So this is why it's shorter. They have a lot of information on like 1 page right here. So let's talk about, I'm still on page 8, by the way. I just clicked the tab for meiosis 2. So meiosis 2 is going to go through a lot of the same things. You're just not going to have another interphase. Okay, so you're skipping interphase. You're going to have step 6. All right, and I'm also out of like divorce references because I don't know. I guess I just don't know any. I'm not really sure why I'm out. I just am. So you're going to start off with prophase. Okay, so prophase 2. Keep in mind, prophase 1, you had crossing over. Prophase 2 is going to be a little different. So prophase 2, you're actually going to end up having more cells. So now you've split and you've got this cleavage furrow thing, right? So you've got this cleavage furrow thing. Now we're down here, and now we're going to actually split into 2 different cells. Again, we're going to have our genetic material cross over some more. So this is more crossing over, but what's going to happen is you have crossing over, but you have half as much stuff now as in prophase 1. So in prophase 1, you guys had a lot of stuff because you guys were just getting married, getting together, or whatever. So you have a whole lot of stuff in prophase 1 up here, right? But in prophase 2, now that you split apart, you guys have less stuff. It still crosses over, but it's your own stuff. So it's really your own stuff crossing with your own stuff. So like your own plates and dishes are just crossing around, doing whatever. That's because you guys are separate. So it's just half as much stuff as in the original one, because in the original one you were living together, in this one you're living separately. All right. I do have more divorce references. If this is hitting like close to home and whatnot, I really apologize. I don't mean to like make light of this. It's just that this is a really excellent analogy for meiosis. Oh, by the way, this is meiosis 2. I know I didn't label it, but you guys probably caught on to that. All right, so step 7, you're going to end up with, again, so after prophase comes metaphase, or the middle phase. So metaphase 2. And during metaphase 2, they are going to line up in the middle again. But remember, you still have half as much line up in the middle. All right, same stuff is going to happen. It's just going to happen again. So if you've memorized meiosis 1, you already know meiosis 2 for the most part. So they are going to line up in the middle again. Keep in mind, it's still only half the material. All right, now we're going to get step 8. And we're going to talk about anaphase 2. So just like after metaphase, you had anaphase, we're going to have anaphase number 2. And then now we're going to, again, have our homologous chromosomes separate. Homologous. So homologous just means, similar structure, because they're all, in this case, they're not going to be tetrads. In this case, they're just, so instead of this thing is no more, we're not doing this, they're going to split this apart, and you're going to end up with, one thing like that, whatever, going one way, and then one thing going the other way to the other side. Okay, so now keep in mind, there's not really a cleavage furrow yet. I just wanted you to know that I meant to draw 2 cells. Essentially, they're going to pull separate. All right, homologous, separate. Again, same things. All right, so this is kind of like you're splitting up with half your stuff. I don't know. I'm kind of running out of. I don't know why you would ever divorce with yourself, but the idea is now that you've got half your stuff, you're losing another half of your stuff. Okay, so let's move on to step 9. And step 9 is just going to be your telophase 2. So step 9 is going to be, and is that on screen? It's kind of on screen. Okay, and you got telophase 2. And it's just going to split again. More cleavage furrows, so more cleavage, and more splitting. More splitting. Notice now that at the end of all of this, you're going to end up with, how should I explain this? You're going to end up with even less, all right? So you lost half your stuff here, you lost half your stuff here. But remember, so how this works is like this, all right? So in interphase 1, you and your significant other, you guys just build up a bunch of crap, right? In prophase 1, you're going to get that crossing over. So now you're living together, all your stuff is getting mixed up. Okay, so you got way more crap than you needed when you were single and when you guys were, living separately, but now you're living together and suddenly you have a whole lot of stuff. So you had to organize that stuff before divorcing, and then you had to separate the stuff. So you had like way more stuff. Originally you had 2 people's worth of stuff. All right, so there's 2 people's worth of stuff. There's 2 people. Let me write this in the marker just as an example. So you have 2 people worth of stuff. All right, but down here at telophase 1, you end up with 1 person worth of stuff. And now each of those 1 persons are going to have all that 1 person stuff, and it's going to cut. Now you have half of a person's stuff. So this is how you end up with your gametes that have half the chromosomes. You start off with 2 different people living 2 separate lives, having 2 things of stuff, so 2 people's worth of stuff. At first telophase, you half that. So each person ends up with just 1 person's worth of stuff. But for whatever reason during meiosis 2, you decide to split again. I don't have a very good example of this. This is like, I don't know, you're living on your own. You're like, man, I just need to live free. So you get rid of a lot of your material. I don't know why. Some people go over these weird things. So, maybe you just reduce your stuff. But anyway, you end up with half person stuff. So just keep in mind that you end up with half as much stuff as a regular person during the second weird divorce with yourself thing. Again, that was a really bad example, but it should make sense. Let me know if it didn't. Just call me. All right. So I know this is going to be a long tutorial, but this is a very, very long topic. So that is meiosis 1 versus meiosis 2. Let's go ahead and take a look. I'm moving on to page 10. All right. So page 10 goes over gametogenesis. Let's talk about gametogenesis. They should talk about spermatogenesis and oogenesis here. They haven't yet, so I guess we'll just talk about it. Oh, they do, because they have the picture for it. They actually just hold on. I'm gonna check page 11. Okay, good, you're good. So gametogenesis. We're gonna move this back into a regular position. Oh, that's right. I moved my camera closer. My bad, guys. There we go. So gametogenesis. Let's go with the screen. Just for the record, this Genesis word, Genesis just means like birth or something like that, or creation. Just like Genesis is the first book of the Bible, so that was when the earth was created. So Genesis is the beginning or the birth of something. Gametes, you already know what those are. Those are those half chromosome things, right? So this is just essentially this is saying the birth of sperm slash ovaries. That's really all gametogenesis is. So how are sex cells created is essentially what they're asking. Alright, so I'm going to go ahead and move on because they're going to talk about the actual types of, there we go. Spermatogenesis is going to be our first one. We're going to do this one in blue. You guys even see the difference? Yeah, a little bit, kind of. If I write big enough, it's pretty obvious. Spermatogenesis, I think that's how to spell it. Spermato. Yeah, that looks right. So spermatogenesis. This is going to be only in guys because only guys create sperm, right? So this is how guys' sperm works. What's going to happen is that you don't need to memorize all of these little names that you see on page 11 on the right. You really have to remember this. So what's going to happen is you start off with twice as much stuff, right? It's going to break down in mitosis into similar things. So this is mitosis. All right, so still 2N, still 2N. This is mitosis. This is a big deal. All right, because your body always starts off with mitosis. Mitosis is the first thing. You guys can't see that. There you go. So mitosis is the very, very first thing, and it's just going to be separating things into clones. All right, so these are exact clones. But now we're going to move into meiosis because now we have sperm to create, right? So we're going to split this one up. We're going to change it to green here. No, you guys can't see. Purple. All right, so we're going to move over here, and we're going to end up with two normal people. So this is like that divorce portion. You end up with two normal people, okay? And then now you're going to do it again, and you're going to split up, and you're going to get haploid gametes. All right, so these are haploids. There you go. So this is meiosis right here. This is meiosis right here, and then you're going to mitosis again. Alright, so you end up with half as much stuff as the original up here, but you end up with 4 of them because technically I should have drawn it for this one too. You'll end up with more like this. Spermatogenesis again is just a process that you end up with 4 of these things. Alright, so in sperm you end up with, for every one sperm, you end up with 4 more identical sperm. For every one cell, rather. You end up with 4 identical working sperm. Okay, so all 4 of these are all good. They're just sperm. Any one of them could create a child. That kind of thing. All right, so this is. Oh my gosh, I'm so sorry, guys. I know this is off screen. 4. There you go. So you end up with 4 of these through meiosis. You end up with 4 working sperm. Okay, all 4 of them are all good. We're gonna go ahead and move on. I'm gonna talk about urogenesis. I know I didn't leave that on camera, so here's the time to pause if you need to pause. I'm gonna go ahead and move on. And we're gonna talk about hopefully oogenesis next. So this is essentially the female version of there we go, good. So I was kind of hoping that this is how it would work. So we're gonna talk about oogenesis now. This was a little less obvious than spermatogenesis. So with spermatogenesis, I was able to easily say like sperm, and you guys know what sperm is. Oogenesis has a much fancier name, but again, genesis just means like the beginning or the birth of something. Oo stands for like ovaries. So this is how we create ovaries. All right, and ovaries are going to work a little bit differently. You are going to start off with that 2N. What color did I use? I used blue last time. Let's use blue this time. So here's your ovary, or I'm sorry, here's your regular cell. It's got 2N, and it's going to be called an oocyte, but you're not going to worry about that. It's going to break down, and it's going to give you 2 N's, right? And and then N. And then what's going to happen is that it's going to split up into one mature ovum. This is a big deal. So this is a mature ovum. It's bigger already. And then you're going to get, let's use a purple, you get like little ones. Okay, this isn't really to scale. They really shouldn't be that much smaller. But you're going to end up with a lot of little ones. They all have the same genetic material, so they're all N. They all have half as many as the original. It's not like you have less stuff or anything like that. But only one of your ova are actually mature and ready to reproduce at a given time. The other ones just kind of chill out and whatnot. So you end up with a lot of these, and they just mature whenever they are ready. So like all of these little things, they're just chilling until you hit puberty and whatnot, and that is when you'll start to actually have these splitting up and the creation of a bunch of different eggs. All right, so that's why children can't get pregnant, because you have to wait until puberty for your ovaries to start actually producing ovum. So these ovum are your ovaries; these are your eggs, essentially. You'll have one mature egg and then backup eggs. So as you shed and whatnot throughout life, you'll end up with more eggs and more eggs that are mature and whatnot. But this is how each split works. Okay. So the difference between, get this on camera if I can. Kind of. All right. Well, they're kind of on the same screen. Spermatogenesis and oogenesis. The big difference is that with sperm, all your sperm are completely ready and completely mature to fertilize at any given time. Your ovaries, on the other hand, you only have one mature ovum at a time. That is why it's like significantly harder for one woman to bear many eggs, many children. So like women cannot have a single woman can't have like 100 children. It's just not going to happen. But theoretically, a man could easily have 100 children with however many women. So it's kind of interesting that way. But that is the difference between oogenesis and spermatogenesis. All right, so that's a big difference, big deal. Let me go ahead and move on because that's really all about these two things. I'm sorry this video is starting to get really long. This is just a really involved chapter. Oh, okay. They actually break down spermatogenesis and oogenesis again on page 13. I don't think I'm too worried about it, so I'm just going to move on. 14 has a question, so let's skip it. Genetic variation is pretty simple. Page 15, they're going to talk about genetic variation. All these things we've talked about, this meiosis thing, this is why we have genetic variation. It's specifically because of, let's see, where it's because of during this process, and again, this is going to be like, landscape mode, you guys can kind of see it, sort of. I think that's all. It's not really all. Well, it's not really all on screen, but you get the idea. It's this crossing over part that's a really big deal, and then you never know with anaphase, they can separate in any way. When you mix them up all in the middle, you guys can argue what goes where. So like the house doesn't always go to the wife, it could go to the husband, whatever. It all depends on the situation. So you never know where things are going to separate. That's called the independent assortment of chromosomes. Okay, so independent assortment means you don't know what trait's going to go where. Crossing over is that stuff here. You never know what gene is going to get mixed up with what. So like which plates are going to mix up with what bowls. You really don't know. It's just going to happen as you're living your life. And then random fertilization really means this. You get to pick your mate. So like females and males, you guys pick. So like when you pick your mate, that determines the genetics. Nobody says you have to date. Well, I mean, some people say you have to date or marry whoever, but the idea is you can pick your mate. Therefore, it is random mating. We don't like it's not a calculated thing. It can be calculated, but it's not. All right. So we already discussed this on page 16, 17, and 18. Look like they're talking about what all these things are. We've already talked about that. So I don't have to talk about that anymore. Random fertilization, again, so I'm not going to break these down. They are on the next 3 pages, so 16, 17, 18 of your tutorial cover these things. But random fertilization, again, you can pick whatever mate. And, you never know what sperm is going to get over there. You think it's like the strongest sperm. It's kind of random. It really is just a sperm is going to get to the egg first, and that's going to be what happens. Right? So random fertilization really means that like you can pick your mate, and then you also don't know it's sperm, so you just never know. It's just not going to happen. Crossing over, any genes can switch up during that crossing over part. You never know what colors you're going to end up with. So like I drew purple blue. This could have easily been like orange, blue, purple, green, whatever. Who knows? So you don't know with crossing over. And then with independent assortment, it really means that when you're splitting up your stuff, depending on how you argue it, you could end up with whatever stuff. Okay, so that's this entire chapter. I know this was a long tutorial. If you have any questions, feel free to ask. I will be producing more soon for this particular chapter, so just give me a little bit of time on this one. I'm actually done for the day, so yeah. Feel free to contact me if anything comes up. Good luck, and let me know what happens.